Hello guys, it's my wonderful pleasure to have you guys again with me, and today we shall be rounding off the paper 1 Gcse question compilation, and you should check my other 3 videos on this. This is the last part. All right, it says which 2 products are formed when lipids is broken down, and that will be fatty acid. That'll be fatty acid and glycerol. All right. The question I want to ask you right now is what forms amino acid and what forms glucose? So when protein is broken down, it is broken down into amino acid, which is what the body cells can absorb. When carbohydrates, which can be starch or glycogen, when these are broken down, I mean the same glycogen, this way they are broken down, it's broken down into glucose, which is the form in which carbohydrate is being absorbed. So these are the absorbable form because it's a bit of digestion, as it were. All right. Moving on. One model used to explain enzyme action is the lock and key theory. The diagram below shows a model of the theory. Okay, ESES. So the E means the enzyme and the S means the substrate, as it were. So explain the lock and key model theory of enzyme action. Use the information above. Well, we can say that so the substrate bind to the active site of the enzyme. So the enzyme is the lock and the substrate is the key. I forgot to add that. Then this forms an enzyme substrate complex. All right. So this, we've talked about this now. This is the second point. So the third point is the enzyme hydrolyze, or you say breaks the substrate into subunits. Subunits, period. So we can then say that the enzyme, the enzyme is available. The enzyme is available to be reused. So that explains that. So really, all the points you have here, the substrate, so what I did here was to talk about this, for this, for this. Last part is this, really. All right. So the way you'll be assessed here is to talk about each of the diagram. All right. I didn't talk about lock and key because it's been established already. The only thing I said was, okay, we added to say the lock was the enzyme, which is the binding site here, and the key is the substrate, as it were. So this is how it's meant to be answered. There are many different types of enzymes, lipase in the human body. Lipase is an enzyme that digests lipids, or we say fat. Why does each different type of enzyme act on only one type of lipid molecules? This is because enzymes are specific to a substrate, or you say enzymes have complementary substrate. That simply means the substrate that can fit into its active site. All right. Students investigated the presence of starch and glucose in the leaves of geranium plants. This is the method used. Place 2 identical geranium plants on the bench near a sunny for 2 days. Okay. After 2 days, leave one of the plants there for another 2 days again. Then place the other one in a cupboard where there's no light. Okay. Remove the leaf from each of the plants. So that means you have 2 plants as it were. So one is kept close to the window, permit that window the way I've drawn it that way to allow it to see the sun. So this is one plant as it were. Then the other plant is kept in the cupboard after 2 days. They were both there initially anyways, but this one is now kept in the cupboard after 2 days. So we're expected to take a leaf from each. So let me paint the leaf, just nothing big, the leaf. So we are taking this leaf and we're taking this leaf. So that means we've taken, so we're meant to say crush each leaf and extract the liquid. So we're meant to take leaf from plant A and from plant B. Test the liquid from each leaf for glucose and starch. Okay. Don't forget the fact that one is still kept beside the sun or beside the window so that light rays can come in, and the other one was taken away. So what does that mean? What's the question here? Describe how the students would find out if the liquid from the leaf contain glucose. Well, here you just, if you can remember food test, you're going to use Benedict test here. So the liquid from each leaf will be added, or no, let me put it this way. Let's start this way. Benedict solution will be added to the liquid from each leaf. That's the one kept beside the window and the one taken away. Okay, then if glucose is present, the liquid will turn brick red, or just red. Let's say brick red. Let me say at this point, in reality, you can check my video on YouTube. It's there. Just check food test, Benedict test. The color can be green, it can be yellow, it can be orange, it can be brick red. Now, the difference from green to this is based on how much of glucose there is. So if there's very little glucose in the fluid or the solution you are testing for, is going to be green. If it is very high, like there's high level of glucose in that is going to be brick red. So, but for sure, the normal thing you just say is brick red, really. So that shows that glucose is present. If glucose is present, the liquid will turn brick red. Then if glucose is absent, the color remains the same. And the color of Benedict is blue. All right. So, or we can say light blue. Let me just say light blue. So what does that leave us here? This was to test your understanding on how to test for glucose. And the way to test for glucose with this is to just use Benedict solution. Then if glucose is present, this is what you see. If it's absent, this is what you're going to see. That's what that is. Then, okay. Describe how the student would find out if the starch, if the leaf contains starch. Well, because this is a 2 mark, I'm going to make it very, because really, if you're going to test for starch, testing for starch goes through different stages, really. But let me first answer this question. I'm going to tell you the real stages of testing for starch. So what you do is iodine will be, sorry, that's too thick. Iodine solution, which is like brown in color, will be added to each of the leaf. If starch is present, the leaf or the liquid, if it's the liquid, or let me say the liquid also here, will turn blue black. But if starch is present, if starch is absent, I mean to say, please, if starch is absent, the color remains the same. So the same color of the iodine remains the same. So you still find the same brown color there. Now, you heard I said, how do we really test for glucose, test for starch? In reality, when you want to test for starch in a leaf, let's say not a crushed leaf now, in a leaf, so you take the leaf from a plant you expected to have photosynthesized, then that leaf will be boiled to kill the leaf. So once you've boiled the leaf, the reason why you are boiling it is to stop all reactions and all of those things. Afterwards, you rinse the leaf, you cool it down, then you put that leaf in a test tube, the same leaf, all right? That same leaf in a test tube that contains ethanol. So this one, you've boiled it here directly, all right? Then here, this is ethanol. So in this case here, you're not putting this. So this is a water bath, all right? So the hotness from this hot water will cause the ethanol to get warm. Then what would that do? That will help you to decolorize the leaf. All right. So afterwards, you bring out this leaf, you cool it down, you rinse it again. Then you add drops of iodine. So if starch is present, the leaf will turn blue, black. But if it's absent, so this is positive. If it is absent, it's going to remain the same. And what's the same color of iodine? Brown, right? It's always, if you see my video on food test, I told you that you should always remember the color of the reagents or the chemical used, because that's the same color you will still see if the result is negative. All right, so this is a quick, if you want to test for leaf, really, like test for starch in the leaf. So if the leaf does not turn blue black, that means starch is not present, but you must have taken it through these stages before you start adding iodine. All right, but I couldn't have written all of these things here for a 2-mark marker question. Okay, it says here, the table shows the students' results. Okay, this is the test on starch, on glucose, on starch. Wow, leaf, okay, strong positive, that's, oh, beautiful. That's brick red, that's going to be brick red. Then it's also in the leaf that is for 4 days. So, 2 days, first 2 days, second 2 days, still kept by the window. But this one, from the leaf kept, okay, the one that is taken to the cupboard. So no starch is present at all, and little glucose is present. Okay, explain why the leaf in the light for 4 days contain both glucose and starch. Interesting. It contains glucose. It contains glucose because it is actively photosynthesizing. Yeah, because it's still photosynthesizing every day. How does it have starch? Because glucose is stored as starch. So it contained starch because it has stored up excess glucose, or let's just excess glucose as starch. This is very important. So which means that this leaf is, because the sun is available, is photosynthesizing, it's also respiring, but it has excess glucose because it kept making up glucose every now and then. Now let's flip the whole thing. Explain why the leaf in the cupboard with no light for 2 days did contain starch. So it did contain glucose but not starch. Well, it's simple. When it's in the dark, so absence of light stopped photosynthesis. Just work with me. So the plant uses the stored starch to carry out respiration. So it uses the stored starch to stay alive. All right. So hence, no new glucose is formed. Rather, starch has been broken down to form glucose. So what it means is this. So let's say this is the leaf like that. It had starch stored up already, but for this leaf not to die, it needs to convert starch to glucose to stay alive. So it's going to finish, so it has converted all the starch it has to glucose. So glucose is present, but starch is not present anymore because it has, like, you store the money in the bank, so you've withdrawn the whole money and you are not working, so you are not getting paid. So since you're not getting paid, you just have to keep spending your savings. And here it says, in fact, it says weak positive. That means if care is not taken, the glucose will be used up for respiration, no more starch to convert anymore, then the liver will start dying, so to say. But if sun were to be available, glucose will be present because glucose is made every now and every day, and starch will also be present because starch is not touched. You're working, you have money saved up, so you don't need to go to the bank to withdraw because you get the money every now and then. Hope that made sense. And this concludes the end of our paper 1 GCSE past questions. I hope this has been helpful. Please do remember to subscribe and like and share, and drop your comment as well. Thank you.